Second phase is the payoff phase. After the investment phase, so there comes the second phase, which we name as payoff phase. In the payoff phase, our body is going to get the profit, is going to get the energy after the investment. So, in the investment phase, we have two molecules of glyceraldehyde 3-phosphate. And in the payoff phase, what happened with this glyceraldehyde 3-phosphate, the chemical reaction number 6, and in this glyceraldehyde 3-phosphate, it will be converted into 1,3-bis phosphoglycerate, and other name is BPG. We can also abbreviate it as BPG. And in this, what will happen? In this, there will be the dehydrogenation reaction, and the enzyme will be named as glyceraldehyde 3-phosphate dehydrogenase. This reaction is very important for the glycolysis. Why? In this reaction, there will be, we can say that there we will conserve a few amount of energy by adding the phosphate to this molecule, and this phosphate, it will be inorganic phosphate. It will not come from the ATP, but it will be an inorganic phosphate, and simply the main purpose of this reaction is to conserve the energy in the phosphate, to conserve the energy by forming a bond through the help of or with the help of this phosphate. So, number one, there will be the addition of inorganic phosphate, and number two, there will be the formation of NADH2. It will be formed by from the NAD positive, and this NADH2, it is also we can say that an energy carrier molecule, and it has the electrons. In the basic foundation years, you have learned that the electrons are the source of energy, and what our body is going to simply store some amount of energy in the form of these electrons in the NADH2, and the few amount of the energy, it is being stored with the help of this inorganic phosphate, and at the end we have a molecule of 1,3-bis phosphoglycerate from glyceraldehyde 3-phosphate. And at this level, this reaction is clinically very significant reaction. This reaction is also involved in the arsenic poisoning. As we know very well that arsenic that exists in two forms, the trivalent as well as the pentavalent form. So, the pentavalent form of arsenic, it will compete for this phosphate, and as a result, when there will be the arsenic poisoning, we cannot, our body, or in other words, we can say that the cell or the body, it will not be able to conserve the energy in the phosphate by attaching it to this glyceraldehyde 3-phosphate and converting it into the 1,3-bis phosphoglycerate. Why? Because arsenic that is going to compete with this inorganic phosphate for this enzyme, and when this arsenic, it will compete with this inorganic phosphate, and at the end it will become attached to this enzyme, and this enzyme will not be able to add another inorganic phosphate into this molecule, and we will not get our product. We will only get 3-phosphoglycerate, not the 1,3-bis phosphoglycerate. So, such type of mechanism of action is named as arsenic poisoning. This is very important. And in the next chemical reaction, what will happen? We will get the energy from this inorganic phosphate, because we conserved the energy that we were unable to get So we conserve the energy into this molecule, and in the next chemical reaction we are going to get that energy back in the form of ATP. So this is 1,3-bis phosphoglycerate, and this will be converted into 3-phosphoglycerate. And simply the enzyme here, the enzyme which is involved, named as 1,3-bis phosphoglycerate kinase enzyme. And what this enzyme is doing, it's simply getting the phosphate back and converting an ADP, one molecule of ADP, into ATP. This reaction is again very significant. Why? This chemical reaction is named as, this process is named as substrate-level phosphorylation. Why it is named as substrate-level phosphorylation? Because you can see that this substrate is directly involved in the formation of energy molecule. So that's why we name it as substrate-level phosphorylation. Is that clear? So this reaction is very important. After that, this 3-phosphoglycerate in the reaction number 8, this 3-phosphoglycerate, it will be converted into another molecule which we name as 2-phosphoglycerate. Here simply there will be, we can say that the change of position of phosphate molecule. In this molecule, the phosphate is attached at the carbon number 3, but in this the phosphate, it has changed its position from carbon number 3 to carbon number 2, and the enzyme involved will be mutase. So in the next reaction, what will happen? That is the reaction number 9. In the next reaction, this 2-phosphoglycerate, it will be converted into an alcoholic molecule which we name as phosphoenol pyruvate, and enzyme involved will be Enolase enzyme. And this enzyme, simply it's converting this molecule into alcoholic molecule, phosphoenolic form, and we name it as phosphoenol pyruvate. Then comes the last chemical reaction. In the last chemical reaction, from this phosphoenol pyruvate, we will get another molecule of ATP. And this is also an irreversible reaction. And we have, at the end, we will have the pyruvate molecule, which is a 3-carbon molecule. And the enzyme involved is pyruvate kinase enzyme. And again, here one phosphate is being removed. So we will have ATP from ADP. From ADP we are having a one molecule of ATP. Again, you can see that this substrate is being involved directly in the formation of ATP. So this is also a substrate level phosphorylation. So in this phase, what we have? We have 2 substrate level phosphorylations, and we have one molecule of NADH and 2 molecules of ATP at the end. But remember, at the end of first phase, at the end of investment phase, we had 2 molecules of glyceraldehyde 3-phosphate. So all these chemical reactions, they will operate twice, and as a result of these reactions, when they operate twice, we will have 2 molecules of NADH and 4 molecules of ATP. So at the end, we can say that end of phase, we have 2 molecules of NADH2 and we have 4 molecules of ATP. And in the start, in investment phase, how many molecules of ATP we have invested? We have invested 2 molecules of ATP. So simply we will subtract 2 molecules of ATP and we will get net 2 ATPs. So at the end, after the balancing of these 2 phases, as we have invested in the first phase, so simply we invested the 2 molecules and we subtracted 2 molecules of ATP from the 4 molecules of ATP for investment phase. And at the end, we will have 2 ATPs and 2 NADH2 molecules. And if we convert this NADH2 molecules into the ATP form, then 1 NADH2, it will be equal to 2 molecules of ATP. Remember, normally 1 molecule of NADH2 is equal to 3 molecules of ATP. But as this glycolysis is taking place in the cytosol and the machinery which is involved to, we can say, extract the energy in the form of ATP, that machinery is present in the mitochondria. And this cytosolic NADH2, it has to travel to the mitochondria. And during this travel time, or during this traveling, they use shuttles. When these molecules use shuttle, they lose their gradient, they lose their energy. When they lose their energy, so at the end, we can say that 1 molecule of NADH2 is equal to 2 ATPs, not the 3 ATPs. And this is only the case with the NADH2 of cytosol, not with the mitochondria. In the mitochondria, 1 NADH2 will be equal to 3 ATPs. But in the cytosol, if they are using, there are 2 shuttles, malate shuttle as well as the oxaloacetate shuttle. But if they are using the shuttles, they have to pay to use that shuttle. When they have to pay to use that shuttle, they lose their gradient. These molecules, they will lose their gradient. When they will lose their gradient, so we will say that 1 molecule of NADH2 then, after losing their gradient, that will be equal to 2 molecules of ATPs. So at the end, 2 molecules from here and 2 molecules from here. What total number of molecules? It will be 2, 2, 4 and it will be, as we know very well that there was 2 molecules of glyceraldehyde 3-phosphate and we will have 2 molecules of NADH2. And if we have 2 molecules of NADH2, we will have total 4 ATPs. So here these 4 ATPs and these 2 ATPs, this will be the total 6 ATPs. So at the end of this glycolysis, what is our profit? Our profit is the 6 molecules of ATPs. And how many number of ATPs we have invested at the start? We have invested only 2 ATPs. And by investing 2 molecules of ATPs, we are getting the 6 ATPs, which is the profit of the cell, which is the profit of cell. Once again, if at the end, as we know very well that in this we have 2 molecules of glyceraldehyde 3-phosphate, so we will have 2 molecules of NADH2. 1 NADH is equal to 2 ATPs, and this is only with the NADH of cytosol, not with the mitochondria. In the mitochondria, 1 NADH will be equal to 3 ATPs. But in the cytosol, this will be equal to 2 ATPs. Why? Because of using the shuttle, they lose their gradient, they have to pay to that shuttle system. So that's why they lose their gradient and their energy is less as compared to the mitochondrial NADH2. So we have 2 NADH2, at the end we will have 4 ATPs. And direct ATP, how many number of ATPs we have directly? We have 4 ATPs directly we are getting from these chemical reactions. and 2 ATPs, they will be given back for the investment, and at the end we will have 2 ATPs. And what is the total budget? 2 ATPs plus 4 ATPs, and there will be the total 6 ATPs at the end of this glycolysis. So this is the energy budget, we can say that energy budget of glycolysis. And this is for the body cells, for the eukaryotic cells. But for the prokaryotic cells, the case will be different. And also for the red blood cells, the case will be different. This energy budget, how it will be different? Here, for prokaryotes as well as RBCs. We know very well that in RBCs we have no mitochondria, because these two NADH2, these two molecules of NADH2, they will only yield the energy when they will go to the mitochondria. Otherwise, if they don't go to mitochondria, they will never yield any energy, and they will be converted back into NAD positive. So, in the case of prokaryotes and RBCs, we know very well that they don't have any kind of mitochondria. When they don't have any kind of mitochondria, so we are not getting these 4 ATPs. Then, like this, 2Na DH2, normally it has to give 4 ATPs. But in prokaryotes, there is no mitochondria. When there is no mitochondria, there will be no production of these 4 ATPs. And what is the rest that we have? That is the 4 ATPs. And we know very well that from these 4 ATPs, we have to pay back the 2 ATPs that we had invested in the investment phase. So, 2 ATPs, and at the end, what will be the result? The result will be 2 ATPs. So, in eukaryotic cells, at the end of the glycolysis, we are getting 6 molecules of ATPs, but in prokaryotic cells as well as in red blood cells, we are getting 2 molecules of ATPs, and these 2 molecules, they are enough for one red blood cell to survive 120 days. This energy is enough for this red blood cell for the survival, we can say for survival till 120 days or for 120 days. So, this is the energy budget of eukaryotic glycolysis as well as the prokaryotic glycolysis, or in other words, you can say the aerobic glycolysis as well as the anaerobic glycolysis. This is the only difference between anaerobic glycolysis and aerobic glycolysis. Both types of glycolysis, they take place in the cytosol. But where is the difference? The difference is only in their energy budget. In eukaryotic glycolysis or aerobic glycolysis, we get 6 molecules of ATPs, and in anaerobic glycolysis, we get 2 molecules of ATPs.